Oh my God, you've been on there with 2 hours.
Because it's connected to her computer, it's only one screen, and I'm going to switch it.
Turn my head for a second.
Hello, everybody. Hi. Hi. Hi over there. Those of you lazy enough not to be here. So it's a wonderful group today, and a great idea that came together from our amazing group of instructors for the Nanolab. You saw their exhibition. We're going to also document it really well. And we host our monthly Leonardo Art Science Evening Rendezvous, which started in San Francisco about almost 10 years ago with Piero Scaruffi. And now it's unbelievable. I just came back from Zurich and there was a laser there. It's just popping up everywhere, which is so exciting to see that this field is just burgeoning. Here on campus, we keep discovering new amazing people. And Noah is one of those new amazing people. And I'm not even going to tell you what she works on because she will. And it's super exciting, definitely dealing with systems. So give her a warm hand.
Thank you guys so much for having me. So I'm a biologist and I study collective behaviors in social insects, mostly ants, as you see here. And I contacted Victoria because I thought there is some potential for interface between artists and science here, and we'll see what you think about it. So specifically, I want to show you this. Oh, and look at that, the movie's not working on this screen, so we should have tested it out. Oh, wait, maybe. Oh, well, it was supposed to start. That's unfortunate. It works on my, I think it's a screen resolution thing. Sorry about that. Anyway, if it had worked, you would see ants move a nest from all the way over here to about 30 meters down the road there, while they're carrying their pupae, and so their brood, their babies, and the queens and princesses that are going to fly off in the upcoming mating season. And the nice thing about this movement behavior between nest sites is that it allows us to look at the behavior of the colony in different sites. And so when you go out to the field, what you see is something like a Swiss cheese of nest sites. And some of these nests are active, and others are not. And then you can come the next day, and all of a sudden a nest site that was active is no longer active, but another nearby nest site is active, and so you can infer that this colony moved, and this happens over and over, and sometimes you're lucky and you actually see the move, which is what I was hoping to show you in that video later. And so my question was whether the behavior of the colony changes as it moves between nest sites. And so what I did was I looked at how they recruit to a piece of food. In this case, it's just a piece of apple. And so I quantified how quickly they call other ants to come to this apple, and I waited for them to move. And once they relocated to a new nest site, I came with a big pile of plaster, and I poured it down the nest, and then I dug it, but this is completely staged because I had lots of little helpers to assist me in the digging. And so what I ended up having are these casts, and I brought some show and tell. Actual plaster cast.
Nice.
So what you see here, this is where the ants would come down, and then these are tunnels and chambers that basically the plaster fills in. And no ants are harmed in the process because, as you remember, they relocate between nest sites. And so I wait for them to vacate the nest before I pour this down. So for example, this nest site had 2 entrances here, and the plaster moved further down in this one and this one. And we can actually pass it around.
Oh,
if you
no,
want.
I want a picture of you with that man.
Okay.
That's great.
All right. And so I ended up here. We can pass this around. So it's just basically plain plaster pouring down down the earth. Do we take questions?
I don't know.
Okay.
Not usually,
okay.
no. Never mind. Not until afterwards, mingling.
All right. And so this is what another example looks like. And I actually went and digitized these. So I have the 3D structures of all of these on my computer. So I have about 70 or so of these, which you can then take and print in a 3D printer if anyone wants to do that, in different sizes and combinations and so on. And I use a lot of network theory in my work, and so the immediate thing that I did was quantify these as networks. So here, the nodes are the different types of chambers, and the tunnels are the edges that connect the nodes. And so this then allows a more quantitative approach to explain what these all sorts of architectural features, which I can then compare across nest sites and correlate that with how quickly the ants recruit to the food item. And so I ended up having lots of.
Between the circles and the squares.
Oh, the squares are the circles are edges of the cast, and the squares are actually chambers that I was certain of them being chambers. And so I had lots and lots of these, and I'll just give you a little bit of the findings. And so it turns out that how quickly ants recruit to the food item on the y-axis here is correlated with how well connected the first chamber that they reach when they come from the outside, which makes sense because they have more places to recruit ants from. The more the nest is connected overall, the faster they are at recruiting. And finally, the more robust the nest architecture is to perturbations, the faster they are at recruiting. So in this example here, you can see that this nest, if you break any tunnel, so any of those single blue lines, it'll completely disconnect the areas from one another. But in that other picture over there, you can block a tunnel, and there's still ways to reach basically any chamber from any other chamber. And so that architecture too increases the speed of recruitment. And so interestingly, there was no relationship between recruitment speed and the volume of the nest, which suggests that it's not how many ants a nest can hold, but how it is designed and how it's basically its structure and architecture. So why I think this happens, this is some modern artwork by ants that if you just leave them to walk around for 5 minutes and film them and then track them. And so this is actually an artificial nest that we keep in the lab. This is that first chamber they reach. These are trajectories walking through tunnels that lead into the arena on this side and further down into the nest. This is an ant that didn't move much, each color is a different ant. And so from these pictures, we can then create interaction networks of the ants. And we find that there is a lot of variation among individuals in how interactive they are. So here, each node is now an ant. Each line is an interaction with a different ant. And the color codes how interactive an ant is. So what you see here is that there's a few very red individuals. So they're very highly interactive. These are like network hubs, basically. And then a lot of other individuals, the blue individuals, that are not very interactive. And this network structure facilitates rapid information flow. And so I showed you that earlier that architecture influences collective foraging. And the current hypothesis for why this happens is that somehow the architecture influences how the ants walk around, which influences how they interact with each other, which influences how they can spread information about an interesting source. And so we're now testing this by giving the ants different structures and manipulating them and so on. And I'll have to come back and tell you about that in the future. But for this card, I also wanted to talk about one other small piece of interesting idea here. So I talked about this project with some social scientists in a National Academy meeting a few years ago, and they mentioned this study that just came out then from social scientists in Michigan, where they looked at the architecture of buildings. So this is a floor plan in a building of a scientific department, and they quantified the overlap between paths that different researchers take. And so if here it's a path between some researcher's office and the elevators and the restrooms and so on, and its lab, and this would be another path, they can then calculate how likely they are to cross paths. And it turns out that building structures that facilitate more of these crossing paths or more path overlap produce more collaborative works and more collaborative papers and more collaborative successful grants. And so this brought us to think, well, maybe we can take lessons from ants and lessons from humans, bring them together, and we actually put a workshop together this past October that was funded by the Keck Futures Initiative, where we had 18 people, 6 were biologists, 6 were architects, and 6 were social scientists. We came together in this place called Decision Theater in Arizona at ASU, which is designed for collective behavior, which was kind of a meta for thinking about how architectures influence collective behavior. And we came up with a bunch of questions that we're now working on putting together as a special issue. So we're trying to put together papers on all these topics. So they include things as can we identify general principles that impact both humans and animal systems? Can we harness and mimic animal builders for human structures? How emotions impact interactions between architecture and collective behavior, which is something that biologists don't think much, but it was interesting to get from the architects. Effective architecture and information flow processing, disease transmission, and transmission of health behaviors. And then how do we actually quantify these architectures and their effects? Various questions about the legibility and eligibility of the built environment and how that influences collective outcome, and then bringing these ideas from theory to practice and actually designing spaces that are inspired by these things. And so when I contacted Victoria, I basically thought that my ants produce these nice, potentially artistic features, and we have these nice, interesting connections with architecture, which is clearly more applicable, applied art, and I will leave you to make further connections that you like, and I'll pass it on.
That talk made me so happy.
Yeah.
It's so exciting. It's so amazing to meet you. It's so great. We're going to be finding you. I'm also really happy and excited about Martina Fröschl, who's here. She just arrived from Vienna last night. And she's here for a project that we're just starting to collaborate on called the Noise Aquarium. She works at the Angewandte in Vienna with Alfred Wendel. And Alfred is a chemist who does these amazing animations based on actual biological data. So they capture the biological data and then animate the data instead of imitating the real life, etc. So we're working with some planktons, and she's going to throw it out there for you to get a sense of where we're going. Hopefully this connects. Usually with Apple, you go do this, and it works. Let's see.
Yep,
There we go.
Nice.
Just the magic.
Perfect.
So, welcome, Martina. Yes, no?
Okay.
Okay, cool.
The back way is more
That's
strenuous.
true, that's true.
Do I have one?
No, you just press the space bar.
Okay. So, hello. Yeah, right. Yeah, I will talk about the Noise Aquarium project. We just started in September, yeah, but we are just the starting point. So I will show some of the animals we are doing. As Victoria said, we are doing scans of the animals, like confocal scans, and transferring them to animation. Yeah, and it is a cooperation project between UCLA and University of Applied Arts in Vienna. So I have just nice pictures of our test screening in summer of our first animals, the paramecium and the amoeba at Ars Electronica Center. It was pretty amazing to see the animations the first time on the big screen. So we took a lot of photos and were very astonished about it. Yeah, and we do, as we try to show the influence of sound pollution, of noise pollution on plankton. So I put here a Wiki Commons image of the phytoplankton because I found it really amazing, and it reminds me somehow on these structures we just did at the exhibition, yeah, with the milk and everything. It was somehow like that, yeah, exactly. And now I will show the animals we already have. So they are real, actual models of the scans. I have here the overview of the animals we already have. We will do some more and then mix them together for the installation. So I have everyone featured of the animals. That's the paramecium in black and white and some smaller particle pyramids in the back. Then I have like I tried to simulate the surface, but it's a little too strong, but it looks cool, so I put it also here. and of course I colorized the organs. We also scan the organs, so yeah, it looks cool when it has a little bit more color here. So the next is the actinotroch. It's a larvae of a worm in the plankton, and this is that's the volumetric. It's a volumetric image of the confocal scan. The next is this cylindrospermum or cyanobacter, also a picture of the volumetric scan. And the Noctiluca, it also is a shining plankton creature, and it floats around. It's like a bubble and floats around, and when it gets in contact with waves, somehow it starts shining in the night. It's pretty awesome. You see that? Then that's the head of the Orcoplora plankton animal. It somehow has like eye structure and the organs also inside. Yeah, another picture. That's the original scan. So it's still somehow a little cramped and a little distorted because of the scanning process and the preparation for the scan. And I always try to fix it, but I try to not destroy the animal's scanned structure, and we have like a feedback process with the biologists that we actually stay at with the shape of the actual animal. So then I have the amoeba, both inside and the outer structure. This one is the brain, the center of the amoeba, and the other one is the whole structure with a little experimental shader. I do a lot of shading experiments on the animation. And here we have a close-up of the Capillaries worm and the volumetric scan, so the actual scan, and then my overworked, also a little more color, yeah, than it has really in the real animal. But yeah, here I just play around with the colors and with the shading and so on, and we will decide to have like more look for that fits to all of the animals. Then yeah, and this is pretty my first glance and overlook of the Noise Aquarium project. So I have put some of the websites that I just connected with, of course Victoria and my website, and then the digital art department of University of Applied Arts, and yeah, of course our nice collaboration partner UCLA. Yeah.
Patricia is going to introduce Adam Hogan, who just arrived from Seattle. Welcome, Adam.
Great. Not like I actually need to read from a script because, well, you might not even know who I am. I'm the artist in residence here at UCLA this semester. So for those of you I haven't had the pleasure to meet yet, that's why I'm here. And we had the good fortune of bringing in someone I'm very happy to introduce. Adam is my former student from Washington University in St. Louis. I'm the director of the Graduate School of Art there. Adam is now in the PhD program at DxArts, and he is an artist and filmmaker with a background in digital and lens-based media. He makes films and installations employing minimalist and abstract cinematic forms that explore anthropogenic traces that humanity leaves on landscape. He actively pursues work and theory in new media, hybrid and collaborative practices, experimental sound, ambisonics, expanded photography, experimental film technology, and field studies expeditions. He's currently based out of Seattle, as I said, and I'm very happy to say he's also my DP and cinematographer on the Mutable Archive project, which is an ongoing project which will take years to finish, which means we'll be working together for many, many years, and Victoria is a collaborator on that as well. So, Adam Hogan.
And actually, I'm going to go ahead and jump out of this and over to the two.
It's in Chrome.
Chrome. Okay, good to know. All right. Since I'm a filmmaker, I'm going to play a video in the background while I kind of give my first little spiel. But it makes more sense to look at it that way as opposed to slides, I think. Just a second. Yeah, so like Patricia said, I'm an artist and filmmaker, and I'm fascinated by humanity's geomorphological presence within the landscape. Our appetite for resources, design, and comfort have arguably made us the largest force that sculpts the earth. And my work often takes me to remote locations and requires these long-term engagements within the landscape. And I've truly been privileged to stand in some of the most surreal spaces, exploring our relationship to the earth and what it really means to be human, not as a ecologist or a scientist, but as an artist. And doing so, like firsthand, gives me the ability to try to bring some of that back to the viewer, to try to give them a small glimpse into the change that our ambition can create. So what you're looking at here is actually the Gunnison Bay, it's basically the northern arm of the Great Salt Lake in Utah. And the extreme salinity in that region was caused by the division of the lake into two distinctly separate bodies of water. So there's three major rivers that supply fresh water to that lake. And over time, since those were cut off from the northern portion, the salinity grew to such levels that it basically killed everything off in the lake except for this red algae and sulfur bacteria, which normally kind of has like a pinkish purple hue, but if you hit it just right and you do enough site research, you can get it to where it's this really blood red, vibrant color. So I was able to spend some time exploring that landscape through aerial cinematography, and essentially it was one of the most surreal spaces I'd ever been into. Like I used to think the salt flats were the most surreal space, like the most alien landscape I'd ever seen, until I spent a couple of days and actually about a week out there. I was like, wow, this takes the cake for everything. And it was truly for me a really sublime and very disturbing example of what a single simple intervention can change and how vast of a change that can create. So what you're seeing is actually excerpts from a cinematic meditation from my ECHO project. And ECHO is a series of experimental films, installations, and cinematic meditations that explore our sometimes tumultuous and strained relationship to the landscape. And I try to do that through spaces of contemplation, as opposed to pointing blame or to calling people out or trying to start an argument. I want to give a space for the audience to come, spend time, and to engage with the landscape through the cinematic apparatus and immersion. And now I'm going to close out of YouTube and jump back to the other thing so I can show some more slides. Throw the DxArts logo in there. Right, and
Just go to view screen mode.
screen mode. Got it. Okay, skip ahead. All right, that's going to bring me to the second project I'm going to talk about, actually, and that is, I had the opportunity to do a public work. So I decided I wanted to do a expanded cinema public work. And the title of this project was Roots, and it was commissioned by the Baum Gallery and UCLA in Arkansas for ArtsFest X. And the project uses projection mapping technology to reposition natural images back within the landscape. But it's not like a natural image that, I'm not trying to say this is the exact things that stood on this ground, but I wanted to give the viewer this kind of ethereal look into a natural space. So the project is trying to look at what we define as a developed space versus a natural space, and what kind of a history the landscape has, And I wanted to use expanded cinema to jump into building a temporary monument or a temporary disruption in this public corridor. Let's skip ahead.
Is this in Ballard?
No, actually, this is in Arkansas, but yeah, I would have liked to do one in Ballard, but I have to shoot it differently.
Okay.
So all of the filming for the project, since it was commissioned in Arkansas, a lot of my practice is site research and spending time within landscapes and really getting to know those places, getting my feet on the ground at those places. And so all of the filming for it was done in a wetland, in focusing on the state's rivers, lowlands, kind of wetland areas, and then the forest. So all of the species and geography are appropriate for that site. Not the exact like historical kind of representation, but a dreamlike kind of ethereal, blurry representation of what a landscape is. And the project is actually, just for size reference, the project is, there are 2 rear projection screens, so in person they kind of float and appear to be kind of no rigging since it's so dark. And then the tall one is 23 by 10 and the small one is 18 by 10. And I was actually able to do some really nice pixel density on the project as well. So you could actually, viewers could rub right up against the screen and get this really nice crisp, kind of very surreal image. And then the last piece I'll kind of talk about, which I won't go into super big detail, is actually a ongoing piece that's kind of been in development. Yeah, it's a little out of control there. So we kind of started this piece as a conversation between several of the PhD students up at the UW. And there's this kind of cliche term about film editing, that great edits happen when you blink, which isn't true at all, actually. But I decided, I was like, well, myself and Dan, who's the composer I'm working with on this project, I was like, well, what if I made a film that was only edited when I blink? All the cuts happen as I'm blinking, as I'm doing the performance. And I tried to put this thing together. So we decided to use EEG technology to integrate and build a algorithmic film and sound composition in real time. So the project is titled Prosthesis Memoria for 2 performers in EEG. And it's an ongoing collaboration with Daniel Peterson, who's the blue cap. I'm the red cap. And essentially, we wanted to look at memory, but more so our digital extensions of our memory and the kind of our own personal archives, the kind of things we generate. And as a filmmaker, I have tons and tons of footage from projects, from freelance work, from my own personal work, from all kinds of, I've always got a camera with me. And Dan is a composer, but he works in ambisonics, which is basically a 3D audio technique to build soundscapes. So he has all these beautiful soundscapes that you can reshape and distort in space. In basically a sphere, if you think of it that way. You can move the sound anywhere closer to the listener, further away. And so we use these to reshape the soundscapes that he's working with, mapped to the neural activity of his brain. So some of the sounds coming in will be repositioned based on that, those signals. All right, let me skip ahead. So essentially we use several different signals. I won't go into super detail, but we can talk about it later if you'd like. And we code those details into compositional control variables. And then we use those variables to, in real time, perform the film. Basically, we're isolated. So I can't see the film that's being created. My neural activity is cutting the film, but the only input that I have is Dan's composition. And likewise, he's isolated. He can only see the film. He can't hear the composition that he's actually creating. And some of the things we track are these, like a P300, for instance, which is like a new, kind of a novelty kind of a reference. And so if I hear something new, that'll pull a new shot and decide when I'm going to cut that next. And then we, like I said, I'll go into more detail later if you really want to know. But basically, we coded in several different variables, and we're constantly trying to code in more and get a little more control, a little cleaner signal. And we literally start off the thing by beginning just with our archives. And then as the piece progresses, we actually are networked together so that we can pull moments where there's kind of clarity, where the video and audio can come together and then come back apart and then bring it together. And essentially we do that by, like I said, we're networked together and we create a feedback loop of what signals are happening, what things need to be changed algorithmically. And yeah. So basically, I'm actually going to stop there because I can rattle on about lenses and things and all kinds of stuff for a long time. But I'm going to pass the podium over, and if you want any more information, just let me know. And I want to thank you guys for having me here. It's great. Thanks everybody for letting me share my work with you. Here's my website.
The next slide. There you go. Woo. How exciting. We have Ursula Hayes finally in person. We meet. So it's another amazing, I mean, the thing about UCLA is you could just never stop discovering incredible people. And here's another one, Ursula. Give her a warm welcome.
Thank you. Yeah, it is amazing to be here. Thank you so much. And this is amazing work to look at. It's really, really fun to get to know this in more detail. So I'm faculty at UCLA. I have a cross appointment between the Department of English and the Institute of Environment and Sustainability. And so Victoria and I have been emailing for, I think, all of the 4 years that I've been at UCLA. And it's great to finally meet in the flesh after all these years. And so, and there is an occasion why this is a particularly good moment because myself and a couple of other faculty came here 4 years ago to sort of foster work and curriculum in environmental humanities. And just a month ago, on October 20th, we launched a new initiative called LENS, the Lab for Environmental Narrative Strategies, in the Institute of Environment and Sustainability, which is on the South Campus. So it's a little bit of beachhead for arts and humanities on the South Campus. And our intent with creating this lab, which grew out of a faculty seminar in 2014 to 15 that involved about 20 faculty and a couple of dozen graduate students from at least half a dozen different departments, was really two things. So one was to show how the rather techy apparatus, vocabulary concept for the study of narrative that's been developed in anthropology, history, and literary studies matters for looking at environmental issues in the way that they're being talked about in images and in text. And not just to show, the human dimensions of environmental crisis, but actually to push a somewhat more radical claim, which is to say that really a lot of the environmental crises and challenges that we're currently looking at and that are most of the time conceived of as scientific and technological problems are really at bottom social, cultural, and historical questions, and that they cannot be looked at without having histories, cultures, and value frameworks in mind. So part of our intent is analytical, and then the other half of our work is creative, to really foster and encourage the creation of new narratives. And in both parts of our work, we really put a highlight on cultural difference. I'm myself from Germany, and I remember being quite surprised when I first came to the United States to see just how differently the American environmental movement talks about some of the issues that are supposedly the same in Western Europe as they are here. And this is, mind you, still all in the framework of the so-called West. So I thought to illustrate, both the analytical and the creative parts of LENS, I would show you a little bit of my own works that I've done over the last few years, and that does involve art, literature, and science in a very pronounced way. And then to illustrate the creative part, to just show you very quickly a few of the projects that we featured at the LENS launch last month. So my work over the last few years has been on biodiversity and endangered species, and as you probably all know, concern about the endangerment of individual species has been around since the 19th century. But it did take a new turn in the 1970s when paleobiologists pointed to the importance of mass extinctions. So extinctions of 75 to 80% of existing species at different moments in evolution, and it really changed the course of evolution. And along with that, and in the collaboration between paleobiologists and environmentalists, the rising suspicion that perhaps we were in the process of seeing another mass extinction, the 6th one in the 3.5 billion years of life on Earth, but this time one triggered by human activity. And so that really gave the concern over endangered species a whole new twist, and since the 1980s, the word that we've used is biodiversity to express that concern. And from the 1980s on, you really get this explosion of cultural and artistic as well as scientific work about endangered species. So you get an unbelievable output of nonfiction, of fiction and poetry across different languages. And these slides, I hope you understand, are just tiny, tiny selections. You also get it across a lot of other areas of culture and art. So you get all kinds of toys that have to do with endangered species. You get your chocolate wrappers at Whole Foods. You get artists such as Maya Lin creating a whole website called What Is Missing and doing various kinds of installations around the world. And Maya Lin, as you probably all know, the artist who created the Vietnam War Memorial in Washington, D.C. And so it's really interesting that she uses her art of memorialization, databasing, and nostalgia and mourning now on behalf of endangered species. You have painters, Isabella Kirkland being one example who revives the tradition of the still life to talk about endangered and extinct species. You have lots of photographers, Susan Middleton and Lichtschwager, and even more famously, Joel Sartori, whose work is often featured in National Geographic and other venues. So all of these there's a real explosion of texts and objects and visual representations that seem to me to call out for analysis in its own right. And now when you think about how many endangered species there are, one thing that's really striking about the artwork and a lot of the public discourse is on how few species that discourse usually focuses. So it usually focuses, the discourse usually in the art usually focuses on animals, almost never plants, although they're equally affected by extinction. And within the realm of animals, it usually focuses on vertebrates, very few examples of invertebrates that attract a lot of public attention. And among the vertebrates, it's usually large mammals, such as the ones you see here, gorillas, tigers, bears, pandas, white rhinos, or big birds like raptors or eagles. So conservationists and environmental communications people often recall these charismatic megafauna, the species that attract a lot of public attention. So how do the stories about them work? So as a literary scholar, that was the thing that most interested me in looking at these various texts. To summarize it in a nutshell, what I found is a really interesting narrative logic of proxies. So you start with the charismatic megafauna who are supposed to stand in for species at large. That's the assumption in both the science and the writing and the art. The species themselves, again, are understood to be proxies for ecosystems and biodiversity. And then ecosystems and biodiversity, each of which are actually species, biodiversity, ecosystems, they're all actually concepts that there's a lot of very controversial debate about, even among scientists. But then they become proxies more in general for what we value about nature. I mean, biodiversity is often used in public discourse just as what's great about nature, and it became mixed in the 1980s with our valuation of cultural diversity. And then when you look at the stories that get told in these about these species and the biodiversity that these stand for, what you find is that the endangered or extinct species usually is meant to symbolize something that a particular cultural community, sometimes a tribal one, sometimes a regional one, often a national culture, has lost or thinks it lost at a particular moment in its own history that has to do with modernization. And when you look at a lot of these stories in the developing world, often modernization comes with colonization. So a lot of the times the endangered species comes to stand for what was there before the intrusion of a European power. and that has since been lost or is believed to have been lost. I don't have time now to go into examples of that, but I analyzed dozens of examples from the Tasmanian thylacine to the Honshu wolf in Japan, to the ivory-billed woodpecker in the United States, to the discourse about whale hunting by indigenous and non-indigenous peoples, to illustrate how that general pattern repeats itself even as there are then distinctive inflections. And so that led to a book that just came out a couple of months ago, Imagining Extinction, the Cultural Meanings of Endangered Species. And in the book, I sort of started with looking at these texts and artworks and looking at how they inflect the science, but also how the science of biodiversity itself is inflected by some of the same prejudices and some of the same narratives that also inflect the public discourse. And that, to me, as a non-scientist, was one of the most surprising findings, that actually you think, okay, the public focuses on charismatic megafauna, but surely the scientists distribute their attention objectively across the whole spectrum of creatures. Not so. I mean, if you look at the IUCN Red List of Endangered Species, the most comprehensive and authoritative red list of endangered species in the international realm, it actually has its weight on exactly the same species and taxa that also dominate public discourse, although, to give them credit, they're also desperately looking for more information about mushrooms and snails and mollusks and all of the other,
do they focus on like pandas, or like what's their term?
No, it's just when you look at, what's been evaluated, so
Yeah.
mammals, all 5,500
Okay.
species of mammals have been evaluated. All 10,000 known species of birds have been evaluated, actually several times over. Birds are the most studied taxon. Amphibians are relatively well explored. Reptiles somewhat less so, fishes somewhat less so. But then when you drop down outside of the realm of vertebrates, you have something like 950,000 species of insects. And of those, maybe like 2,000 have been explored. You have 30,000 species of mushrooms. As of my last count, I think 5 or 6 had been assessed in terms of their risk of extinction. So it's really quite drastic. So it's vertebrates and then certain kinds of flowering plants, and the rest is really quite underexplored even on the part of science. So the same cultural investments and the same narratives of decline of nature under the impact of modern societies are actually also very widespread in the scientific texts. So I went from looking at that interface between science and art and literature to other things like red lists of endangered species and biodiversity databases, and Victoria's work actually was very influential in that, sort of looking at how a certain elegiac aesthetic informs a lot of these scientific databases, and then how that databasing instinct actually informs art, such as that of Joel Sartori and several writers who work by way of just enumerations of endangered species. I got interested in endangered species laws across different cultures and how they also reflect that shared concern and different histories of engagement with endangered species. And in the second half of the book, I sort of put the cultural concern over endangered species that expresses itself in science, laws, and art into larger contexts about conflicts between the conservation movement and the environmental justice movement, conflicts between conservation and the animal rights movement, and the framing of these debates now in the debates about the Anthropocene, the idea that we have pervasively reshaped the planet, and that obviously, if it's true, has profound implications for what we want to conserve and why. And it has, that's work that I think has immediate implications for how we think about conservation in the practical realm as well. So for me, the main interest was, what does it tell us about cultural communities? What does it tell us about narrative and its relation to science? But it also has, my contention in the book is that you have to know these stories if you want to do conservation effectively, and I give examples of where and how that's true. So this is sort of one example of the kind of analytical work that we want to do with Lens, where we think about environmental storytelling that's out there with some of the tools that anthropology, history, and literary studies have provided us, and film studies also in a major way. So Lens is about understanding stories and conflicts like this, but it's also about encouraging faculty, students, and communities outside of the university to tell new stories about the environment. So we understand Lens as a sort of incubator for new narratives, new environmental narratives, and new in two ways. I mean, one, really interested in just stories about the environment that haven't been told, things that we don't know about, and then stories of any kind, whether they're new or old, that are being told in new ways. And I thought actually, Adam, your film was quite a beautiful example of how you move away from the typical sort of didactic documentary film into a very different kind of mode. And that's exactly the kind of thing we're interested in, innovative kinds of video and film, graphic novels, animated film, propagation of environmental narratives through social media and so forth. And so at our launch last month on October 20th, so we invited a bunch of novelists, photographers, journalists, and artists to participate, and we had talks and discussions, and also a bunch of art installations. So this is our website, and also we have started up a magazine that has its online magazine that has first few stories up there. So one of the artists we had visit was Valerie Cohen, who together with Michael Cohen has done a book called Tree Lines, which is about bristlecone pines in Nevada. Valerie is a watercolor artist and a drawer, and so she has done a series of amazing drawings of the intricate structures of the bristlecone pines, which actually in their structure remind me a little bit of these plaster sculptures. Jessica Catalina, who teaches at the Department of Anthropology here at UCLA, has done a collaboration with the photographer Adam Nadel on the cultural politics of water in the Everglades. So we had a pop-up exhibit with his photographs and excerpts from her interviews and her comments on the photographs, where they talk about the uses and perceptions of water around the Everglades on the part of Native Americans, farmers, sugarcane corporation officials, government officials, ranchers, and so forth. So Jessica has done hundreds and hundreds of interviews with them, and Adam has done a lot of really, really interesting photography on this. We had Lars Jan's exhibit Holocene's Anthropocene Suite 1 at the Fowler Museum at a raucous party on October 20th. So Lars creates water tanks in different cities around the world, and he has people inside these tanks that perform everyday activities. A woman carrying a basket of apples, a guy trying to deal with a sack of rice. And gradually the water will rise in the tank and will sort of slowly discombobulate both the objects and the people. Now, installing one of those water tanks would have cost tens of thousands of dollars. It was way beyond our budget, but so we commissioned from Lars a video of his installations, which was playing on a square video screen with four sides around the central fountain at the Fowler. And finally, we also had Kendra Sollers and Lauren Strohacker from Tucson, who did one of their Animal Land installations at Sunset Plaza, where the undergrads lived in the dorms 3 nights. And so this again was work that was specifically done for this installation. So what they do is they take short videos of wildlife, then play it in photo negative and in slow motion, and project it on the outside of buildings. And in this case, we commissioned them to just do animals that have been around the LA area, that were once here, could come back. And so here's the barn owls. And they also had an interactive part to this, where the students could submit their own wildlife photos, and they sort of integrated those into their projections on the last night of their performance. And then finally, we screened Marina Zurko's animated work at Mesocosm Wing, Texas. It's hand-drawn animation that's run by a computer algorithm, and the landscape of an oil sinkhole in Texas goes through various day and night and seasonal variations as well as eruptions and other changes. And you can sort of see on one hand the beauty of the monarchs and the ominous element of people who can only move about in the landscape in hazmat suits. And as you saw, that is also what became sort of, with Marina's kind permission, became the image that we use to popularize lens around campus to make it known. So that was sort of the science, art, writing interface that Lens is meant to be an incubator for, and we really look forward to collaborating with many of you individually, if you're so inclined, and with Sci collectively. So really looking forward to that.
Yes. Yeah, it's really exciting, and I think the environmental narratives that are about to start happening.
Yeah.
Jeez. Whew. Right?
Yeah, I mean, there's a lot of new narratives coming out of LA, a lot of stories about the environment from the Latino community that really have not been part of the mostly white environmental movement's conception of things. So that's very exciting.
Frida's like, yeah. And I'm talking also about our new administration and the narratives we're going to listen to. Their ideas about the environment. It'll be interesting. Yes.
So
And
did people see the photo of a blackboard outside a bookstore that circulated on social media today? Our post-apocalyptic fiction has moved over to current affairs.
Left cry.
yes.
That's crazy.
So with that, I will now introduce Mick, who's going to introduce the whole fluid systems exhibition.
Yeah.
This is how are you doing?
It's Olivia, me, and
Okay.
Dan are going to be speaking here.
Yeah, this is, I feel just so incredibly and truly honored to work with you guys, and really to have this team, the art science team, is just phenomenal because it's self-organized. Nobody put out a call or anything. We just came together naturally. And the kind of beautiful work that's coming out is just amazing. So give these guys a warm welcome. Come on.
Dan? Where's Dan? Did he leave? I guess it's us.
He'll come back.
All right.
Okay.
Yes, sir.
Yes, so we're going to speak loosely about our collaboration, which, as I mentioned in the opening, good to see you, Evolutionary, that we were actually inspired by our collaboration through the SciArt Nanolab. So the SciArt Nanolab is an intensive 2-week session for high school students that are interested in the intersection between art and science, which of course the Art Sci Center is all about. And we take over the lobby of CNSI, and we also spend 2 weeks intensively as an instructor group brainstorming ideas, and we were very much focused on ecological systems and fluid systems, especially the collective that's formed organically between Oliva Osborne, Dan Wilkinson, and myself, Mick Lorusso. And so we are going to talk a little bit about the focus that we developed in some of these workshops and what's happening in the show here.
So before you move on, this is a very basic biological fluid system. It's a capillary bed, essentially. And you can see a vein, and an arteriole, an artery, and a venule. Oh, sorry. Yeah. I mean, they can kind of see it. I'm just reading off the screen now.
Yeah.
But essentially, when you first look at this, it's already a fluid system. I immediately thought, what does this also look like? Because we're talking about also in the Nanolab, going from like a micro to macro level. This also reminded me of just an ecological network that we find possibly in nature, but I was also fascinated by the concept of seeing it also in urbanization. So this could be like a waterbed, and then these could be like little sort of inlets or the estuary in a sea, for example. And this was also the theme upon like the works that you saw in the art gallery. So just bear that in mind when we talk about that. But
when we talk about all of these things, you've probably never heard me lecture before, but I do love like the sort of theory behind the word, so the Latin, the etymology of the word, and essentially all of these things, the most fascinating one that I loved actually was networks. And networks, I found out that originally meant wires. So originally it meant just like wiry networks. And then English people decided, you know what, networks actually means something more like a transport system, specifically like a railway or a river. So I thought that was really interesting as well. And it wasn't until 1986, the year before I was born, that they decided to make it something that meant a group of people. And I thought all of those things were super interesting because it means not only a network in biology, it means a network in a transport system, which is exactly what a capillary is, and in this case, like to deliver blood all over the body, but it also means a network of communication between people and a society. So that's the basis of our work, essentially.
Exactly. And our collaboration
Essentially.
started in this way. And I would also like to mention that art-science collaborations oftentimes do revolve around these kinds of fluid systems, interestingly enough. I mean, you think about Leonardo da Vinci and his work on fluid dynamics and the relationship between micro and macro fluids. And Dan Wilkinson. Yes,
So yeah, this was the slide that, I didn't know if this one was in or not yet, but essentially, to break it down, fluid just means something that's like a liquid in motion. A system is an organized network. A network is also an organized system, and ecology just basically means the study of a biological system, but also in terms of the preciousness and vulnerability of an ecosystem. So it's the study of how basically pollution and stuff that we do in our everyday lives can actually affect essentially that ecosystem. And these are like buzzwords that we tend to use in our nanolab, but also in our projects. So if you want to start off with capillaries or...
go for it, Dan.
Yeah.
Would you like to address some buzzwords?
Sure. Capillary, or capillary, is just any really narrow channel. And this is actually the mechanism by which plants bring up water into their bodies. Let's see, the micro and macro. I think it's really interesting. So some of the interactive piece that we were looking at really reminds me of actually looking at Jupiter and the clouds of Jupiter, and especially the Great Eye. And so we see a lot of similarities on the micro scale and the macro scale, from a styrofoam plate with milk on it to the fluid dynamics on a giant planet. Dendrites are basically these branch points in crystal formation, and the sort of the basis of snowflakes. And it's really interesting how a simple angle, right, the angle between the oxygen and the hydrogens in water, can produce these really radical and very beautiful patterns.
And dendrites comes from the word dendron, which means tree.
Yeah.
Interesting, huh?
Cool.
Right. And here we are at the observatory, the eve of my birthday this week, hanging out with Einstein. So we decided to call ourselves the Einstein Club. Anybody wants to join, welcome to participate in a base test initiation rites, which is non-Newtonian fluid initiation, because Einstein, of course, is non-Newtonian. So I think that, you guys want to add anything to that?
It takes a lot of bravery to chuck Newton out the window.
Yeah, it does. Yeah.
But Einstein did it pretty thoroughly.
I put this in because this is very close to my heart, even though I'm a hardcore scientist, as most of you will probably know. I do like to have an outside life, and this one I really like because it's Vincent van Gogh writing to his brother from London, basically. And it says, "Always continue walking a lot and loving nature, for that's the real way to learn to understand art better and better." And then there's a bit of spiel, but I cut that bit out. "Painters understand nature and love it and teach us to see." And from that, I wanted you to see that even though you think there's something in nature that you think, oh, that's really cool and that's really nice, for me, the more I look at it, the more I understand more about myself, and the more I try to understand more about life and the way we act upon things. So I just wanted to put that in there because it's one of my favorite quotes, and roll with it, basically. This is a cool motion that is not going to happen, apparently, because you've turned it into a PDF.
That's right.
And not told me. And,
Yeah.
okay, well, I'm going to pretend that I'm the water particle, which is a first, not a last, apparently. So essentially, some of the stuff that you saw in there is to do with how water interacts with chemicals and things like that. This is just, do you want to load it, actually? Yeah. So, oh, apparently you don't want to see a performance dumps piece. About,
No, start the performance.
so
and then
the
you're gonna see
particles
the
at the
real
bottom move super slowly, right? The ones at the top create this like wave, and as it creates this wave, it creates the particles like this, essentially. And that's the basis of some of our pieces almost, the way that water particles like interact in and out. So the deeper they are, the less they tend to actually interact, whereas the ones at the top tend to get to like mesh together more, and you'll see finally if you pull this up.
Well, I don't want to block your slide here. So here.
Oh, it's
Just
okay,
keep
don't
talking
worry.
to it, okay.
But there's this really
Yes.
cool mathematical sort of equation that scientists came up with, which is like the wave phase, which in itself is quite a cool word buzz. And they worked out some really sort of geeky stuff.
What's the etymology of wave?
Don't know, Amisha, do you want to tell us?
No, I don't know.
I actually don't know that one myself.
I don't know.
Please check it
I don't
out.
know.
Let's check it out.
Never not know.
Well, I think
I
one
know.
of the interesting things is that these little water packets aren't traveling, right? They're just sort of bobbing up and down. So it's really this momentum that's being carried that's moving, rather than individual pieces of water or packets of water. And I'm sure that the animation will bear that out.
Yeah, the reason I didn't know is because it's not Latin, and I'm not really interested in Germanic base. So it's called a waiver. I know, I'm sorry if I offended people. But essentially, there is no reason why it's called wave.
There can't be no reason.
No, there's no reason.
Okay, there it is. It's worth the wait.
It was worth the wait and not my interpretive dance move. It was worth the wait.
It was worth
The wave.
But essentially what you can see here is these particles move super slowly, basically. And then these ones move medium, more and more. As you get to the surface, they tend to move more and more, which is exactly essentially what the pigments were doing as well in the photos that you saw and in the gallery. The closer they are to the surface as well, if you put more movement and momentum to it, the more they pick up. And essentially this is like a wave of motion that goes on, and that's why you see waves in the sea that do that, essentially. And that's a small gift to show what it looks like in case you've never been to the ocean. But also I love this new concept as well of like a capillary wave. So we were talking about capillaries and what they do, but a capillary wave actually is the wave traveling along the phase of a fluid, that the dynamics are dominated by the surface tension, which is exactly what I showed you, but basically capillary waves are known as ripples. Wow, full circle. So essentially, I know, this is just like two bowls underneath a swimming pool, which is actually this exhibit that happened in Santa Monica a couple of weeks ago. The actual bowls are like, in my view, very, mundane and not worth your while. But what's really interesting from this, no offense if you were the actual artist providing those bowls, of course, but what was really interesting were the ripple effects that came off of the pool, in my view, which is the capillary wave. So still playing on the theme of like capillaries and ripples and effects and nature, essentially. I just think it's a cool picture anyway. And then, Mick, do you want to take over this bit?
Yes, so Olivia and I decided to go back to our artistic roots, because Olivia also is a painter, and I also started off as a painter. And so one of the pieces in the show is a media projection of a performance, a painting performance that we devised. And essentially, we're talking about fluid dynamics as a relationship. So we are applying watercolors to these plexiglass or perspex panels, as they call them in England. with with absorbent paper, and they're on a fulcrum. So as we kind of negotiate the space, these fluid dynamics form across the surface. And I think really the piece is really about that relationship. So it's far more interesting to see the dynamics arise in the video as opposed to the final product. So that's how we chose to present it.
But also, if you look at the painting, especially the one on the left, it looks like, oh wait, that's what it looks like. Back to our original piece, sort of like capillary network, and that's what we tried to recreate. We actually had these glue channels, which were obviously man-made, but then the actual liquid itself, the watercolor, created its own channels on the paper, which I thought was very interesting. Dan, do you want to talk about also some of our work?
Well, so one thing I think about is really interesting when thinking about how fluids move in capillaries is this idea of laminar flow. Does anyone know what that means? Anyone have a...
Laminar flow as opposed to turbulent flow is when the surface of a liquid moves smoothly without any kind of perturbations.
Yeah, it's this like lack of mixing. And I think that's really interesting. So if you can envision I'm a leaf and you're a leaf, and we fall into this this river, and we'll say this river is undergoing laminar flow. Don't fall into my liver. It's a bit toxified at the moment. But let's say you're a leaf, I'm a leaf, and we fall into this river, and this river is under a laminar flow regime. That means that as we travel down the river, we look at one another, and we start this far away, and the river may bend and turn, but we always remain that far away, no matter what. Now, turbulent flow is mixing, which means that as we travel down the river, let's say there's a tight bend and we eddy, and we might bump into each other and converge, or might diverge and never see each other again. So this idea of how these fluids move, I think is nicely elaborated in that analogy. And so as you're a red blood cell moving through the blood, you're in this laminar regime where if you're separated from somebody else, you remain separate for a long time. Now, of course, there's points of turbulence, but I think this it is very interesting in terms of how we understand how fluids move in very tight channels. What else was I just talking about?
No, that's pretty much.
That's all I have for capillaries.
Yeah, so I think that's the conclusion of our collaborative talk, and thank you, Art Sci, for this amazing opportunity to show collectively our brainstorming efforts that have occurred over the last 6 months or so. So.
And if you have obviously any
questions or
questions?
How would you enact Brownian motion in a dance?
One brown, I'll get right up there.
Yeah, we also have to get into that.
there you go.
Back to Einstein.
Interestingly, Einstein wasn't awarded the Nobel Prize for special relativity or general relativity, but he was awarded the Einstein Prize, the Nobel Prize for
the photoelectric effect.
For the photoelectric effect, yeah,
exactly.
Do that again. Do the dance again.
More random.
More random. We need music. More alcohol.
More speed, less control.
Right. Good.
So, for those of you who don't know, this group, including Amisha, they are actually instructors for a program that we do in the summer called Nanolab. Rita as well.
Rita.
Rita. And these are actually amazing people who are the teachers of our future and who teach our young students who are ready to go into universities to stop separating art and science. We try to catch them early, so when they go into the universities, they actually demand this. It's our kind of strategies. It's been this year, coming year, 2017, will be 10 years we're doing this. And I calculated we have about 700 youngsters out there who are thinking like this, which is so cool. So anyway, thanks to these guys, because they themselves, you don't know where art ends and science starts and vice versa. And it's just fabulous. So thank you. Anyway, is there anybody here who wants to announce something? What you're doing? An exhibition?
Oh,
Come up here.
yeah.
Come up here so we can.
Slide
We're
show,
streaming.
slide show.
There's more people out there than in
Website,
here.
slideshow.
This is Chris O'Leary. Chris O'Leary.
I'm a former student of Victoria's,
Yes.
went to
I'm
DMA
very
about
proud
10
to
years
say.
ago.
Yeah, wow.
And 10 years ago
Wow.
this year. If you are over in Pasadena near Art Center at the Hillside campus, there's a show called Uncertainty that's about physics and quantum mechanics and brings together artists and scientists, and I have a piece about black holes there. So if you have a chance.
It's amazing, I've seen it.
Yeah, it's an immersive generative animation, and it's quite lovely. And Edward Tufte's in the show, and a bunch of great people. So, and I will present in January about this.
Yes, exactly.
So,
Is that the next
That's the
laser?
the next laser. So please, if you're in Pasadena, come check out Uncertainty.
Yeah. Announcements? Okay, now we mingle.